This lecture is on Chapter 10, Muscle Tissue. What is the muscular system? Muscles transform the chemical energy of ATP into mechanical energy and exert force. There are three types of muscles: skeletal, cardiac, and smooth. Please note that the prefixes myo, mys, and sarco refer to muscle tissue. The three types of muscle tissue: skeletal muscle is part of the musculoskeletal system, cardiac muscle is found in the walls of the heart, and smooth muscle is found in the walls of the hollow organs. Skeletal muscle provides voluntary movement of the skeleton, eyes, and facial skin. Skeletal muscle cells are called muscle fibers. Cells are cylindrical, multinucleated, and striated. Cardiac muscle is found in the walls of the heart, and cardiac muscle produces the involuntary movement of the heart. Cells of cardiac muscle are branched, uninucleate or binucleated, and striated. Cardiac muscle cells are joined by gap junctions called intercalated discs. Smooth muscle, which is found in the walls of the hollow organs, produces the involuntary movement of digestive organs and blood vessels. Smooth muscle cells are small, uninucleate, fusiform in shape, and non-striated. This table shows the comparison of skeletal, cardiac, and smooth muscle. This is from another textbook called Marieb, and I love this illustration, so hence the chapter 9. But you can see here that with skeletal muscle, it is attached to the bones, or some facial muscles will attach to the skin. And you can see the single, very long, cylindrical, multinucleated cells, obvious striations pictured. Moving over to the cardiac muscle, the walls of the heart are where we'll find that. And the illustration shows that cardiac muscle has branching chains of cells that are uni, meaning one, or bi, meaning two, nucleate, and there are also striations. Note those intercalated discs that are darker in appearance. Those are the gap junctions that allow ion exchange. Smooth muscle to the right is a single unit muscle in the walls of the hollow visceral organs other than the heart. There are also multi-unit muscles in intrinsic eye muscles, airways, and large arteries. This has to do with the nerve input. The cell shape of smooth muscles are single and fusiform, meaning pinched at both ends, uninucleate, and there are no striations present. This chapter is going to focus on skeletal muscle, but still all muscles do share these following special characteristics. The muscle cells are excitable, meaning they can respond to nervous stimulation. They have contractility, meaning that they can shorten and produce tension or force. They have extensibility, meaning they can stretch. And they have elasticity, meaning they can return to their normal shape. So let's move now to the skeletal muscle in particular. So the functions of the skeletal muscle include producing movement, maintaining posture and body position, stabilizing joints, generating heat, and also providing protection of abdominal organs in particular. The gross anatomy of skeletal muscle is as follows. The skeletal muscle is a discrete organ, and it is made of several kinds of tissues. They will, of course, be muscle fibers, but also blood vessels, nerve fibers, and connective tissue. The illustration shows the muscle attached to the bone by a connective tissue called the tendon. The tendon is continuous with the epimysium, a connective tissue that surrounds the entire muscle. The paramysium and the endomysium are connective tissues deep within the muscle. The fascicle is a bundle of muscle fibers, And the muscle fiber is pictured as a cylinder coming out of the fascicle. And then the blood vessels are shown strewn throughout the interior of that muscle. We'll look at the nerve fibers as we move into the physiology. So let's take a look at the connective tissue layers a little more in depth. Remember, the epimysium, paramysium, and endomysium were just mentioned in the illustration prior. The epi means that it is going to be around the entire muscle. Peri is within the muscle deep and it's surrounding fascicles. Endo is the deepest of the connective tissue layers. And recall that mys means muscle. So epimysium surrounds the entire muscle. It is a dense irregular connective tissue that surrounds the muscle like a leather sheath, and it is continuous with tendons. And the tendons themselves are dense regular connective tissue, but they merge into the epimysium as dense irregular connective tissue. The paramysium surrounds the fascicles of the muscle fibers, and it is also dense irregular connective tissue, so it will be tough fibrous sleeves that protect and support the bundles of muscle fibers within. The endomysium is going to surround the individual muscle fibers, and this is a delicate tissue, areolar connective tissue, and its major purpose is to electrically insulate the skeletal muscle fibers. The neat thing about the connective tissues is not only do they electrically insulate the muscle fibers, pictured down here, the bottom right. So the muscle fiber is the actual cell of the muscle, and the endomysium is going to be surrounding that, electrically insulating them, helping to ensure that the excitation is going to remain within those cells and travel the appropriate direction. And then the paramysium, which surrounds the entire fascicle there, that dense irregular connective tissue, it's going to be connected to the other paramysia that are within that muscle, and those are all going to ultimately connect to the epimysium, which binds the entire muscle into a unit, an organ. And so when the muscle fibers contract and then the paramysium contracts as a result, it ends up pulling on the epimysium, which pulls on the tendon, and then that is what helps to move the bone and produce the entire process of movement. The attachments of skeletal muscles include the origin and the insertion, as well as direct and indirect attachments. The origin is going to be the part of the tendon attached to the immobile bone when a movement is produced. So the origin will not move. The insertion is the part of the tendon that attaches to the bone that is going to move during movement. So in this illustration of the biceps muscle, the origin is going to be the scapula and the insertion the radius. So when flexion occurs and pulls the forearm towards the arm, the radius, the insertion, is going to be moving towards the origin on the scapula, and the scapula will stay still. So the origin stays still and the insertion is what moves. With direct attachments, the muscle will be fused to the periosteum of the bone, and that is pictured here with the deltoid muscle fusing to the clavicle and the scapula. And then with indirect attachments, like the tendon, it's going to be a cord-like structure that will insert on the bone. So let's look at tendons in more detail. Tendons are cord-like, and then there's a type of tendinosis kind of material called an aponeurosis, which is a sheet-like. And the tendons and aponeuroses are composed of dense regular connective tissue, and they ultimately will pull on the bones when the muscles generate force. There's fascia that describes the collagenous network of membranes deep to the skin. Deep fascia surrounds the epimysium of a skeletal muscle. It is dense irregular connective tissue. Superficial fascia is found within the subcutaneous layer of the skin and is adipose tissue. So far between the tendons, aponeuroses, epimysium, paramysium, and deep fascia, this is all collagen. Remember that collagen is the most abundant protein in the body, and there are several types of collagen. I believe there might be 5, And types 1, 2, and 3 I know are in supplement form most commonly, and these are all going to help to protect the skin, muscles, joints, hair, nails, bones. This illustration shows the tendons as cord-like and the aponeurosis as sheet-like, seen on the upper left-hand side of this PowerPoint slide. And then on the upper right-hand side of the PowerPoint slide is the connective tissues, those superficial and deep fascia. So the superficial fascia, you can see, lies between the skin and the underlying organs and is primarily adipose tissue, and it's the subcutaneous layer or hypodermis. And then the deep fascia is going to be bound to capsules, tendons, and ligaments. It forms dense connective tissue, and it is a strong fibrous internal framework. There's even subserous fascia between the serous membranes and deep fascia, and this is areolar tissue. But the collagenous network, a lot of folks will have like tension in the fascia, and that can cause problems. It can cause knots and movement issues. Also, you've heard of plantar fasciitis, which is the inflammation of the fascia in the plantar region or the bottom of the feet. And then in women particularly, fascia between the superficial fascia, there's cords that can form, collagenous cords, and that connects the skin to the deeper fascia, and that is what can end up causing dimpling as the spaces in between, fill with subcutaneous fascia or adipose tissue, and that can lead to cellulite. There is a surgery that can be done that goes and snips those cords to smooth out that particular region of tissue there. It's not a permanent surgery though, because those cords will reconnect, but it does last for a little while if cellulite is a big problem in that area and the woman or has money to do it. And the reason I say women is because it's a woman issue, cellulite. So that was the gross anatomy of the skeletal muscles. And now let's look at the microscopic anatomy. So recall that skeletal muscle cells are called muscle fibers. And during embryonic development, hundreds of embryonic stem cells will fuse together to form one skeletal muscle fiber, resulting in a long cylindrical multinucleated cell. And when the cell is matured, it's not going to be able to fully regenerate. If it is torn, it will scar. There will be stem cells left over to help maintain the muscle fibers during the lifetime, but this is a tissue that at this time cannot fully regenerate. So let's take a look at the part of the muscle fiber, which is the skeletal muscle cell. The plasma membrane of a muscle fiber is called the sarcolemma. So remember, sarco is one of the prefixes that means muscle. So sarcolemma is the plasma membrane of a muscle fiber. Deep to the sarcolemma is the sarcoplasm, and this is akin to the cytoplasm of a cell, because remember, the muscle fiber is a cell. So the sarcoplasm will contain large amounts of glycosomes and myoglobin, and this will provide energy for the muscles during contraction. And then the usual organelles are also present, as well as many nuclei, and of course a lot of mitochondria. Myofibrils are long rods of protein within the muscle fiber, and these are going to be filled with myosin and actin myofilaments. So the myofibrils are going to be pictured here. They are the long rods of protein, and the actin and myosin myofilaments will be contained within the myofibrils. Surrounding the myofibrils will be the sarcoplasmic reticulum, which is a modified smooth endoplasmic reticulum. And the purpose of the sarcoplasmic reticulum is to store calcium ions, because calcium ions are going to be integral to the contraction process. T-tubules are going to be found abutting adjacent sarcoplasmic reticulum, and this is going to be the way that the electrical impulse that comes from the motor neuron can enter deep into the muscle fiber and cause the release of calcium from the sarcoplasmic reticulum. So the myofibrils are composed of contractile proteins called myofilaments, which are the thinnest of the units within the muscle fiber. So myosin proteins make up the thick myofilaments, and actin subunits make up thin myofilaments. Actin will be surrounded by the supporting proteins called troponin and tropomyosin. Tropo means in the way, and tropomyosin is going to be in the way of myosin. We'll explain that in a moment. The contractile unit of the muscle fiber is called the sarcomere, and the sarcomere is located between what are known as Z-discs. Z-discs zigzag, and that's what their name comes from, and they anchor the thin myofilaments or actin, forming the borders of the sarcomere. The center of the sarcomere is going to contain the M line, and I think of M as middle, and this is what anchors the thick myofilaments, the myosin, and this forms the center of the sarcomere. There will be a potential space in the center of the sarcomere called the H zone. I think of this as the holding zone. So looking at a myofibril, the myofibril is going to be filled with the myofilaments, actin, and myosin. In this illustration, the actin is going to be a thin red line attached to the Z-discs. The Z-discs are in blue. And then the myosin are going to be looking like they're thick yellow rods. They are thicker than the actin. So the Z-disc forms the boundary of the sarcomere as illustrated here, and the actin is attached to the Z-disc. The myosin is in the center of the sarcomere attached to that M line, forming that very middle spot. And then that potential space is the H zone, or that holding zone. The sarcolemma is shown surrounding the entire muscle fiber, which is the muscle cell. So remember, the sarcolemma is the plasma membrane of the muscle cell. The sarcoplasm is going to be interior, and here's a large nucleus pictured, as well as several mitochondria. Notice that the muscle fiber, or the muscle cell, is filled with myofibrils, and these are going to be those rods of protein, and the rods of protein are made up of myofilaments. So it goes muscle fiber, which is the entire cell, muscle fiber, and then myofibril and myofilament. Here's another view of a sarcomere, just looking at one myofibril. So the myofibril has the numerous myofilaments within it, and the Z-disc is going to be a type of protein that forms that boundary of the sarcomere. And here the actin is illustrated in blue, attached to the Z-discs. The myosin is in dark red, and it's in that center there, anchored to the M line. And then that H zone is the holding zone. In the illustration at the bottom of figure 9.2, this is also from that Marriott book. You can see that the Z-disc here is anchoring those actin myofilaments. And then the myosin is in the center, anchored by the M line. And then there's your holding zone. You might notice that little coil of yellow protein that's called the elastin, and that helps the sarcomere return to its normal position after contraction. This shows the sarcomere with the sarcoplasmic reticulum laying over it. So in the very top there's the myofibril, and no sarcoplasmic reticulum is on there, so you can see the Z disc exposed, and the actin, and the myosin, and the M line, and the H zone. Notice that where the T tubules are, that lies over the Z discs. And then the sarcoplasmic reticulum are going to lie over the interior middle of the sarcomere. So it will overlie those myosin and the M line and the H zone. And this is all very on purpose. So once more, now let's take a look at the actin and myosin just without any of the other features of the sarcomere. And so we can see the actin is made up of little subunits. They look like little beads. So those are the actin subunits. And then the myosin, being the thicker myofilaments, are actually composed of many proteins that are going to be twisted and end in these globular-like heads. So looking over at tropomyosin and troponin, I'd mentioned that tropo means in the way. And so tropomyosin is in the way of myosin. So what happens here is this long protein, this tropomyosin, is covering these divots, which are active sites. And these divots are the perfect shape to fit into the myosin heads. So during the process of contraction, the myosin heads will fit into those actin divots, those active sites of the actin. And the heads of the myosin will actually walk the actin subunits towards the center of the sarcomere, filling in that H zone. But when contraction is not occurring, the tropomyosin is going to block those active sites. Tropomyosin is going to be in the way of myosin, and that helps to shut the system down so the muscle can relax. Troponin is a yellow protein that is going to help hold the tropomyosin in the appropriate place. So when tropomyosin is covering the active sites of the actin, it will keep it steady. And then when contraction occurs, troponin will change shape and move that tropomyosin out of the way. This illustration shows what's called the sliding filament model of muscle contraction. And in picture 1, up at the top, is a fully relaxed sarcomere. And when it's fully relaxed, the H zone is open, the M line is exposed, and the Z discs are shown, and then there is, some light banding and dark banding. Notice that even when relaxed, the actin and the myosin slightly overlap. And that makes sense because they have to overlap in order for the myosin to attach to that actin when contraction occurs. In the illustration number 2 on the bottom of the screen, you can see that contraction had occurred and the myosin heads were able to attach to the divots on the actin, those active sites, and walk the actin subunits towards the center of the sarcomere. And at the end of contraction, the actin myofilaments have filled in that H zone. So how does that occur? Well, in order to figure that out, we have to look at skeletal muscle physiology. Let's start by discussing something called the action potential. The action potential is the result of a predictable sequence of electrical changes in the sarcolemma as a result of a nerve impulse. So a motor neuron is going to send an impulse that will travel through the sarcolemma. And in order for an electrical impulse to occur, the sarcolemma has to be charged. So even at rest, the sarcolemma is charged with a membrane potential of -70 millivolts. When the action potential occurs, it changes the membrane potential from the resting -70 millivolts to a +30 millivolts. And this is what causes the electrical impulse to occur, this switch in charges. The steps involved in the action potential are as follows. In step 1, there's a generation of a localized depolarization at the neuromuscular junction. This is called an end plate potential. Whenever you see the term potential, think electricity at this point. So step 1 is the generalization or the generation of a localized depolarization at the neuromuscular junction, and this is called the end plate potential. Step 2 is depolarization and generation of the action potential as sodium ions enter the cell. This is what causes the inside of the sarcolemma to become a positive 30 millivolt. During step 3, or repolarization, potassium ions will exit the cell, and this restores the sarcolemma to its initial membrane potential. The illustration shows the action potential. Before the action potential occurs, the sarcolemma is a negative 70 millivolts. And this negative 70 millivolts charge is just on the inside of the sarcolemma in the cell. So it's on the interior. During the end plate potential, the channels are going to be allowing for the influx of sodium ions. Sodium ions have a positive charge. And the end plate potential is going to bring the membrane potential to a threshold of negative 55 millivolts. Once negative 55 millivolts is reached, this is kind of like the trigger of a gun and the bullet will fire. The bullet firing is going to be that depolarization stage. At this time, sodium is going to flow in mass, and it will spike that membrane potential to a positive 30 millivolts. This will end up propagating all the way down the sarcolemma into the T-tubules. After the depolarization wave has occurred, the shot was fired, repolarization is the next step to follow. In order to repolarize the membrane, the sodium channels have to close, so no more sodium can influx into the cell, and potassium channels are now going to open. Potassium is going to leave. Notice that potassium is positive as well, and potassium will exit in large amounts, and ultimately the membrane potential is going to bypass or surpass that negative 70 millivolts, and it'll get even more negative than negative 70. This is the stimulus that initiates the potassium channels to close. Through natural leakage channels of sodium and potassium, as well as the sodium-potassium pump, the resting membrane potential of negative 70 millivolts will be reestablished. This illustration shows the action potential along with the sodium and potassium permeability. So recall, in the initial stage, the negative resting potential, the resting membrane potential of negative 70 is maintained. So the only way that the end plate potential is going to be reached is if there is an electrical stimulus coming from a motor neuron. And so at that neuromuscular junction, when the motor neuron is firing, it's going to open sodium channels, and the sodium is going to start to flow in. As the sodium is flowing in, the interior of the sarcolemma is going to become less negative at that neuromuscular junction, and it's going to reach that end plate potential of negative 55 millivolts. Once it reaches that negative 55 millivolts, that gun is going to fire, and the depolarization wave is going to be initiated. And the sodium influx is going to increase dramatically as sodium flows in through the channels into the cell. And this is going to spike that action potential at a positive 30 millivolts. After the action potential depolarization has passed, then the sodium channels are going to close. And notice here that sodium permeability is going to drastically reduce. And now the potassium channels are going to open, and potassium permeability will increase. Rather than coming into the cell, though, potassium is leaving the cell. So we're leaving positive ions, they're going away, and the repolarization wave is going to bring the membrane potential back to its negative state. It's even going to surpass that and go more negative than negative 70 millivolts. Once that occurs, that's the stimulus for the potassium channels to close. And notice that the potassium permeability is going to continue to decrease. And then over time, with natural leakage of the sodium and potassium into and out of the cell, as well as the sodium-potassium pump, the resting membrane potential of negative 70 millivolts is going to be reestablished. So this is an illustration of the motor neuron, and the motor neuron is going to initiate from the spinal cord. So this is the part of the motor neuron that will be in the spinal cord, and the axon, which is that long tail, is going to extend down the motor neuron and be traveling in whatever nerve is going to connect with the muscle at the neuromuscular junction. So the three main events that occur in a muscle contraction is going to be step one, the excitation of a muscle fiber at the neuromuscular junction. So this is when that end plate potential is going to be really reached. And if it is, then step two occurs, which is excitation, contraction, coupling. So the electrical signal in the form of an action potential is going to travel down the sarcolemma into the T-tubules. This will stimulate the release of calcium from the sarcoplasmic reticulum and initiate step three, the crossbridge cycling. So this will occur when the calcium binds to troponin, moves tropomyosin out of the way, and initiates the sliding of the myofilaments. Let's look at the steps in more detail. So step one, the motor neuron will stimulate the muscle fiber at the neuromuscular junction. All right, so number one, the nerve impulse will arrive at the axon terminal of a motor neuron. Number two, voltage-gated calcium channels will open. Number three, calcium will enter the motor neuron and cause the release of a neurotransmitter called acetylcholine. Number four, the acetylcholine will diffuse down the synaptic cleft and will bind to receptors on the sarcolemma of the muscle fiber. Number five, electrical events, meaning leading to the end plate potential, will lead to the generation of an action potential. Number six, after the action potential has been propagated, the acetylcholinesterase is an enzyme that will break down the remnant acetylcholine. So this figure shows the steps of generation of the action potential. So in number one, the action potential arrives at the axon terminal of a motor neuron. So this is the axon terminal of the motor neuron. In number two, voltage-gated channels open and allow for calcium to enter the axon terminal. Number three, calcium entry will cause the synaptic vesicles containing acetylcholine to release their contents by exocytosis. Number four, acetylcholine, the neurotransmitter, will diffuse across the synaptic cleft and bind to the receptors on the sarcolemma. This will open them up in step five, allowing for the influx of sodium into the muscle fiber, and potassium can exit as well. But we're really getting a drastic influx of sodium here. And this is going to bring the neuromuscular junction to that end plate potential and propagate the action potential down the sarcolemma. After that occurs, 6 comes into play, and the acetylcholine effects are terminated by the enzymatic breakdown by the enzyme called acetylcholinesterase. So once the action potential is propagated, excitation, contraction, and coupling can occur. So number one, the action potential propagates along the sarcolemma and down the T-tubules. Number two, calcium ions are released from the sarcoplasmic reticulum. Number 3, calcium binds to troponin and removes the blocking action of tropomyosin. And number 4, contraction begins as a crossbridge cycle occurs. So here is the excitation contraction coupling. So you can see that the acetylcholine has come down, diffusing down the synaptic cleft, binding to receptors on the sarcolemma. The end plate potential has been reached, and that was negative 55 millivolts. Once the end plate potential is reached, then the wave of depolarization goes down the sarcolemma. So remember, depolarization is when the sodium channels are open and sodium is flowing into the sarcoplasm and spiking the action potential to a positive 30 millivolts. Once the action potential has passed, then the sodium channels close and repolarization can occur. And this occurs when potassium channels are open, potassium is leaving the sarcoplasm, and that is reestablishing the membrane potential of negative 70 millivolts. It will surpass that negative 70 and what we call hyperpolarize, and that closes those potassium channels. Once that occurs, then the natural leakage of the sodium and potassium, as well as the sodium potassium pump, can bring the membrane back to negative 70 millivolts at rest. So the sarcolemma is going to carry that action potential all the way down. The nerve ends up coming in and binding to a muscle fiber, generally around the middle of that muscle fiber, and then the action potential can spread proximally and distally down that muscle fiber. As it's doing that, it's going to go deep into the muscle fiber via the T-tubules. All along the T-tubules will be more voltage-gated channels that will open as a result of the action potential, and that's how that calcium is going to be released. So here's those steps. 1, the action potential is propagated down the sarcolemma and down the T-tubules. All along those T-tubules and the sarcoplasmic reticulum will be calcium channels, and the voltage gated channels respond to action potentials. So far, the calcium channels in the neuron, the sodium, and the potassium channels in the sarcolemma are all called voltage-gated channels. And so that action potential, that switch of the membrane potential at rest of negative 70 up to the threshold of negative 55, all the way up to that positive 30 millivolts, the whole purpose of that is to open these particular voltage-gated channels. So in this case, as the action potential is traveling down the T-tubules, it's opening the calcium channels, and this allows the release of calcium into the sarcoplasm. And at this point, you know where it's going. It's going to bind to troponin, and that troponin then is going to undergo a shape change and move tropomyosin out of the way. And now the little divots on the actin, those active sites, are exposed to myosin. Myosin can now attach to the actin and start walking those actin subunits towards the center of the sarcomere. And that's what initiates the contraction. So that's why it's called excitation-contraction coupling. So this picture shows the sliding filament model of contraction that I had mentioned prior. And to remind you of that, what's happening here is the sarcomere shortens because the myosin walks the actin towards the center of the sarcomere. So they're sliding over one another. And that's why there's this overlap even at rest of actin and myosin. And there you see again the illustration number 1 shows a fully relaxed sarcomere of the muscle fiber, and illustration 2 shows a fully contracted sarcomere of a muscle fiber. Crossbridge cycling is what occurs as the myosin is walking the actin towards the center of the sarcomere. So let's take a look at that before we leave the physiology. So during this crossbridge cycle, the attachment of the myosin heads occur on the actin filaments. And the crossbridge cycle will continue as long as calcium and ATP are present. There's a power stroke where the myosin head pivots and pulls that thin filament towards the middle of the sarcomere. Then the crossbridge has to detach. So the myosin head will detach from that little divot, and it then recocks so it can attach to another actin divot. So here's the crossbridge formation. The troponin has moved the tropomyosin out of the way, and the myosin can attach to the little divot there, that active site found on the actin. The second is the power stroke. So notice that the myosin is pivoting towards the center of the sarcomere, pulling that actin with it. In order to make another crossbridge and continue the walking process, then the myosin has to detach. And then it recocks so that it can attach to another actin divot. So here it all is together, and I can describe what's going on with the ATP. So take a look at step 1 there where the crossbridge formation has occurred. Notice that there are 2 molecules attached to myosin, ADP, which is adenosine diphosphate, and an inorganic phosphate, illustrated as PI. So ADP in red and PI in purple. So notice that they are attached to the myosin head as the crossbridge formation occurs. So in order to pivot the myosin head and make that power stroke work, the ADP and inorganic phosphate have to be removed from that myosin head. So there'll be an enzyme that will remove those from the myosin head. So as those are getting removed away, that causes the pivoting of the myosin head in step 2, moving that actin towards the center of the sarcomere. In order for the myosin to form another crossbridge, it has to detach from the one that it just moved. And so to do that, a new ATP is going to come in and attach to myosin. So that attachment of the ATP is going to detach that crossbridge in step 3. Then we have to recock the myosin head, basically re-energizing it. And the whole point of ATP is to give energy. And the way it does that is through hydrolysis. So the bond between the terminal phosphate and that ADP is broken. And now you can see that ADP and inorganic phosphate are attached to that myosin head. The energy that was released from breaking that bond between ADP and inorganic phosphate is what recocked the myosin head. So now the myosin head is free to go back to step 1 and form a crossbridge formation with another actin. And then the ADP and PI will be removed, and the myosin head can pivot forward, moving the actin towards the center of the sarcomere. Then another ATP can come in and attach to the myosin, which detaches the crossbridge. And then once that ATP hydrolyzes into ADP and inorganic phosphate, that re-energizes, aka recocks the myosin head so it can attach to another actin. And it's going to continue to walk the actin towards the center of the sarcomere until contraction has completed. So it takes a lot of energy for this to occur, because every time that myosin pivots and then detaches, an ATP is involved. And so ATP is the only source used directly for contractile activities. In the muscle, the ATP stores that are already available are depleted within 4 to 6 seconds. So there's no way that that can sustain muscle contraction. Therefore, ATP has to be regenerated quickly by one of 3 methods, or all 3 are working together. So in the method number 1, it's called direct phosphorylation of ADP by a molecule called creatine phosphate, abbreviated CP. The other way is anaerobic glycolysis. And then the third way is aerobic respiration, which is going to provide the most ATP. Let's look at step 1, or the method 1, the direct phosphorylation first. So this happens during the first 15 seconds of vigorous exercise. The need for ATP will soar higher than the available stores can sustain. Luckily, creatine phosphate can be stored in abundance, and the creatine phosphate will donate its phosphate to ADP. Muscle cells can store up to 3 times more creatine phosphate than ATP. This reaction is reversible, and inorganic phosphate can be reattached to creatine, and thus creatine phosphate can be regenerated easily. This illustration shows the creatine phosphate and an ADP merging together, and there's an enzyme called creatine kinase that will take the phosphate off of the creatine and attach it to the ADP. And so you're left with the products creatine and ATP. And this can provide duration of energy for 15 seconds. And then the anaerobic pathway can take over, and this occurs when large amounts of ATP are needed for moderate periods, like 30 to 40 seconds of strenuous activity. It occurs at 70% of maximum contractile activity. So when you're, sprinting really fast, or, throwing a punch, or throwing a ball, or jumping, that will cause the muscles to bulge, which will compress blood vessels. This causes oxygen delivery to be impaired and pyruvic acid to be converted into lactic acid. Together, the creatine phosphate and anaerobic pathways can sustain intense muscle movement for up to 1 minute. Lactic acid is a byproduct of the anaerobic pathway, and this has been in controversy because does it cause the muscle pain? Does it not? Lactic acid will diffuse into the bloodstream, and the liver, kidneys, and the heart can use it as fuel. Lactic acid will eventually be converted back into pyruvic acid or into glucose by the liver. Then it is released back into the bloodstream, or it will be converted to glycogen as storage for later use. Lactic acid is an acid, so it can lower the pH of the muscle, causing a little bit of acid burn. So there may be something to the lactic acid causing a little bit of muscle strain, but we'll talk about exercise later. So here's the anaerobic pathway. You can see that glucose is the primary energy source here, and glycolysis translates as the breaking of glucose into 2 molecules of pyruvic acid. And so the pyruvic acid then has 2 fates. It can either go into the mitochondria for aerobic respiration, but it has to have oxygen to do that. So the second fate is there is no oxygen, and so it's converted into lactic acid, which will be released into the blood. Glycolysis will produce 2 ATP per glucose, as well as the lactic acid, and so it's only going to be able to provide energy for about 60 seconds or a little more. The aerobic pathway is going to be the major pathway for our energy provisions. Because creatine phosphate stores are limited and the anaerobic pathways only generate 5% of the ATP that muscles can use, the aerobic pathway will take over and provide 95% of the ATP. It sustains muscles during long bouts of activity as well as during rest and moderate activity. The aerobic pathway will occur in the mitochondria, requires oxygen, and involves a sequence of chemical reactions that break down nutrients. So glucose is the one that is generally mentioned when teaching the aerobic pathway, but fat and protein can also be used and can be broken down to drive the synthesis of ATP. So the fuels are stored glycogen, blood-borne glucose, even pyruvic acid from the glycolysis as mentioned earlier. If the fat is being broken down, it will be free fatty acids. Proteins are only used if not enough glucose is available, such as during a fast, and that can produce wasting of muscles. So one needs to be careful not to actually burn the protein as fuel. It's more desirable to burn the glycolysis or the glucose and fatty acids. So here's the picture of aerobic cellular respiration. The glucose in the blue is going to be broken down by glycolysis into 2 molecules of pyruvic acid. And the pyruvic acid can go into the mitochondria for aerobic respiration. Fatty acids can also go into the mitochondria, and then there's the amino acids from protein if necessary. So thus, the glucose, fatty acids, and amino acids are what we call energy-yielding nutrients because they all three can be used for energy. Again, primarily we want to use amino acids, though, to synthesize proteins and build things in our body rather than burn them as energy. They're really priceless. But glucose and fatty acids are the preferred fuels. So between those three, if oxygen is present, 32 ATP can be gained. And so that's a significantly more amount of ATP than what was seen with creatine phosphate and glycolysis alone. Creatine phosphate gives you 1 ATP, glycolysis gives you 2, and aerobic respiration gives you 32. And it's astonishing the amount of ATP needed to power and sustain muscle contraction. So aerobic respiration is really the only way. All right, so during short duration exercise, within the first 6 seconds, ATP stored in the muscles is used. And then in the next 10 seconds, ATP is formed from creatine phosphate and adenosine diphosphate. And then within 30-40 seconds to the end of exercise, glycogen stored in the muscles is broken down into glucose, which is then oxidized to generate those ATPs. So that's a real short, intense burst of exercise. And then in prolonged duration exercise, which can, go for hours, the ATP is generated by the breakdown of several nutrient energy fuels by aerobic pathway. This pathway uses oxygen released from myoglobin or delivered in the blood by hemoglobin. When it ends, the oxygen deficit is paid back. So the body's going to tailor and use, a mixture of all of these fuel sources. If the muscles become fatigued, then they have the physiological inability to contract. And this can occur when ionic imbalances interfere with the excitation-contraction coupling, or if prolonged exercise damages the sarcoplasmic reticulum and interferes with calcium regulation and release. Now, muscle fatigue can heal as the person rests over a matter of days. The oxygen deficit occurs because humans use only about 25% of the oxygen they inhale. So 75% of the oxygen is exhaled as waste. So whales use 90% of the oxygen they inhale, and I always put that in there because, a whale is a mammal and it's breathing air, but they just come up and, take a quick breath and can go down into the water for 45 minutes or more. And that's because they have, way more efficient myoglobin and hemoglobin usage and stuff than humans. But again, we're living in air, so we have the luxury of being able to waste 75% of our oxygen in our exhalations. So anyway, that can build up an oxygen deficit. So extra oxygen is going to be needed after exercise for the replenishment of the oxygen reserves in myoglobin. Accumulated lactic acid must be reconverted to pyruvic acid or glucose. Glycogen stores have to be restored, and ATP and creatine phosphate reserves have to be restored. So even anaerobic activity can accrue an oxygen debt because oxygen is necessary to restore the above resources. And as one becomes more physically fit, the oxygen deficit is easier to recover from. Only about 40% of the energy released in the muscle activity is useful as work. And so again, we're wasting about 60% of that energy it's given off as heat. But that's important though, because that helps to maintain body temperature and warm us up when we're cold. And if we're exercising and it's hot outside, then of course we'll sweat. And that helps to prevent dangerous heat levels. This illustration shows a human who is warm-blooded and holding a scorpion, which is cold-blooded. And so the infrared is showing the heat that is radiating off of the human. And notice that where the human is holding that scorpion's tail, see how it's red? So the heat is being transmitted from that human to the scorpion. That's not me. I wouldn't be holding that thing. All right, so there is a condition called muscular dystrophy, and it is a group of inherited muscle-destroying diseases. Muscles will initially enlarge due to fat and connective tissue deposits, but the muscle fibers will soon atrophy and degenerate. Duchenne muscular dystrophy is the most common and severe type. It is inherited and sex-linked, meaning that it is carried by females and expressed in males. 1 in every 3,600 births might have this, and the expression is a lack of dystrophin. So the disease will progress from the extremities upward, so from the arms and legs upward, finally affecting the head and chest muscles and cardiac muscles. Victims become clumsy and fall frequently, and usually die of respiratory failure in their 20s. So the reason that the females don't express it as much as males do is because if it's sex-linked, it's on the X chromosome. And if, a girl has 2 X chromosomes and one of them does not have the muscular dystrophy, she will, be a carrier, but she herself will not have muscular dystrophy. But if that X chromosome that is the muscular dystrophy gene gets passed down to a boy who has just that X and a Y, then he will express the muscular dystrophy. So let's talk about dystrophin a little more. I mentioned it in the previous slide. So dystrophin is a protein that helps stabilize the sarcolemma, which is the plasma membrane of the muscle fiber. So without dystrophin, the sarcolemma will tear during contraction. This causes electrolyte imbalance and inflammation, and contractile myofibrils, the actin and myosin, are damaged. Macrophages and lymphocytes will accumulate around the damaged tissues to clean them out, and over time the damaged muscle fibers will actually undergo apoptosis and die. And remember how I said that once you are grown out of that developmental stage, if those muscle cells are dead, they're not going to regenerate at that point. So unfortunately, there's no cure for Duchenne muscular dystrophy, but the steroid prednisone is the only drug at the time of this slide that improves muscle strength and function. I'm sure that researchers are working on it diligently. So on the left of this slide shows normal muscle tissue with healthy dystrophin. That's pictured in green. And then on the right is a patient with muscular dystrophy, and you can see how the dystrophin is decreased and the sarcolemma is going to then degrade as well. So these illustrations show a healthy muscle on the left versus the diseased muscle on the right that has been broken down. Now let's look at some physics. So these are going to be the review principles of muscle mechanics. So the same principles will apply to the contraction of a single fiber and a whole muscle. Contraction is going to produce tension on a load, and contraction does not always shorten a muscle. In isometric contraction, tension develops, but the load is not moved. Thus, the muscle is not shortened. In isotonic contraction, tension develops and the load is moved. So the muscle is shortened. So isometric and isotonic, both of the iso means equal. So with isometric, I just kind of think of it as like equal pressure. So you can hold an object basically, but you're not moving it. So basically if you're just, holding like a baby in your arms, but not moving that baby, that's an isometric contraction. If you're pushing against a wall, but you're not moving the wall, but you still feel that tension, that's an isometric contraction. Anytime you do a contraction that moves the object, that's isotonic. So the force and the duration of contraction will vary in response to stimuli of different frequencies and intensities. And the stimuli is going to be coming from the load that is being placed on the muscle, and then the resulting electrical stimulus coming from those motor neurons to help you bear that weight or move that weight. So a motor neuron has what's called a motor unit. One nerve will serve a muscle, and it usually enters the center of the muscle. Within a nerve are going to be numerous neurons, thousands of them, if not more. So a motor unit will be a motor neuron and all the muscle fibers it supplies. So the motor neuron will branch out, all those little axon terminals will branch out, and it will connect to several muscle fibers. So one motor neuron can have several neuromuscular junctions and excite several muscle fibers, causing the end plate potential to occur and the action potential to be propagated. And in a small motor unit, the motor neuron will, only innervate 4 or so muscle fibers, and that will move very fine muscles found in the fingers and the eyes. However, there will be large motor units where a motor neuron will branch out and innervate hundreds even of muscle fibers in weight-bearing muscles like the thighs and the hips. So the small motor units are obviously going to fire much easier than large motor units. The muscle fibers in a single motor unit are not clustered together, but are instead spread throughout the muscle. And this is convenient because stimulation of the motor unit will cause a contraction of the entire muscle. The more muscle fibers that are engaged, the more powerful the muscle contraction is. Motor units in a muscle will prevent fatigue because they usually contract asynchronously. So as one motor unit is tiring, another motor unit will fire. This illustration shows 2 motor units. Number 1 is pictured in purple and number 2 is pictured in red. And you can see that motor unit number 1 is a motor neuron that is branching and synapsing with 2 muscle fibers. Motor unit number 2 is branching and synapsing with 3 muscle fibers. So axons of the motor neurons will extend from the spinal cord to the muscle. There, each axon will divide into a number of axon terminals that form neuromuscular junctions with muscular fibers scattered throughout the muscle. So as motor unit number 1 will fire, those 2 muscle fibers attached to it will fire simultaneously. Eventually, they're going to get tired. They're going to use up their ATP and glycogen or fatty acid stores for energy, and they're going to need to relax. So then motor unit number 2 is going to fire, and those 3 muscle fibers will contract and take over. And that's what that asynchronous firing was talking about earlier. The muscle is going to produce more force based on the tension placed on it. And so let's talk about the response to the change in the stimulus strength. The threshold stimulus is the stimulus strength at which the first observable muscle contraction will occur. So that's where you're going to reach, that negative 55 millivolts at the end plate potential and stimulate the action potential. Beyond the threshold stimulus, a muscle will contract more vigorously as the stimulus strength is increased above the threshold. So again, that's going to be dependent on the load being placed on the muscle and the motor neurons that are firing. So contraction force is precisely controlled by recruitment. So multiple motor unit summation will occur, which brings more and more muscle fibers into action. Motor units with larger and larger fibers are recruited as the stimulus intensity increases. So as the load is heavier and more motor neurons are firing from the brain and the spinal cord, then more motor units will be recruited to help bear that weight. So here you can see at the very top, the stimulus threshold 1, 2, and 3 don't reach the stimulus. 3 is almost there, but we're not going to see muscle fiber twitch until 4. So you've got to pass that stimulus threshold in order to initiate the motor units that are going to really cause any noticeable movement in muscle contraction. And then by the time you get to the maximal contraction here, that's not going to occur in a normal muscle. That can occur in a lab. And you can see that all the motor units are going to be excited in that particular muscle fiber. So that's not in nature. That couldn't happen really. That would cause you to break your own bones. And so, speaking of when a person gets shocked, that electricity that's coming into the body is traveling down the motor neurons and initiating muscle contractions. And so it can be so forceful that it can cause the victim to be thrown by the jolt. And oftentimes, the victim can actually, suffer joint and bone injury as a result of the intense contraction caused by that exterior electricity coming into the body. And as you know as well, that can also cause heart defibrillation to occur and a heart attack. Okay, so in the first illustration here, the green motor unit 1 will be recruited first with the small fibers. These are going to be the most excitable ones. And then with more tension, motor unit 2 will be recruited that has medium fibers that can bear more weight. And then if the tension continues to increase, then recruitment will continue to occur where larger fibers will be recruited to help bear that weight. So a muscle twitch is a motor unit's response to a single action potential of its motor neuron. Usually the muscle undergoes graded responses because a single twitch is not enough to generate enough force to move an object. And you know what a muscle twitch is because it's literally just one quick movement of the muscle and then it goes back to rest. Some muscles will twitch faster than others. The eye muscles twitch very fast and do not sustain contractions for longer periods. Calf muscles will twitch slowly because they do need to sustain contractions for longer periods. So what that means, the twitch is the time from contraction to relaxation. So the quicker the twitch, the faster the muscle can relax. Here's the extraocular muscle of the eye. See how quick that twitch is there, so it will relax very easily. This is the gastrocnemius and the soleus of the calf muscle, showing that it takes a lot longer for it to go to rest during that muscle twitch, which helps it sustain a contraction for a longer period of time. The nervous system will achieve greater muscular force by increasing the firing rate of motor neurons. If two identical stimuli are delivered to a muscle in rapid succession, the second twitch will be stronger than the first, and this is called summation. And summation occurs because the stimulus is delivered before the muscle can completely relax. This occurs because calcium is already in the sarcoplasm, ATP is already being used, and the muscle is already partially contracted. So this is different from motor unit recruitment. This is occurring within the same muscle fiber, the same motor unit. So the summation can result in a process called tetanus, and there are two. There's incomplete and complete. So incomplete tetanus occurs if the stimulus strength is held constant and the muscle is stimulated at a faster rate. So the relaxation time between twitches will decrease, concentration of calcium in the sarcoplasm rises, wave summation increases in a sustained but quivering muscle contraction. And so that is what's going to occur during really intense muscle movement, like, heavy weightlifting and such. Complete tetanus is very rare. It's a sustained muscle contraction at maximum tension. And as mentioned earlier, this is going to occur like in a lab. You're not going to see this in an actual person. So this is the wave summation. And if another stimulus is applied before the muscle relaxes completely, then more tension will result. And this wave summation results in unfused or incomplete tetanus. This is complete tetanus that you're not going to see in a person. It will be in a lab. The force of contraction is determined by the number of muscle fibers recruited. So the more motor units that are recruited, the greater the muscle force. The size of the muscle fibers. Larger fibers will produce greater muscle force because there'll be more protein in there and more contraction. And then the frequency of the stimulation. If a muscle is stimulated rapidly through a rapid succession of motor neuron firing, then contractions will become stronger and more vigorous, eventually producing that tetanus. Muscle tone is the constant, slightly contracted state of all muscles, and it's due to spinal reflexes that activate groups of motor units alternately in response to input from stretch receptors in the muscles. This keeps muscles firm, healthy, and ready to respond. So skeletal muscles are generally voluntary when you're actually going to do voluntary movement. But yes, there is some involuntary reflexive muscle tone that is coming from the reticular formation of the brain that's maintaining that muscle tone. So as mentioned earlier, muscle fibers are going to help to influence the contraction of the muscles. And so muscles will vary in how fast they can contract and how long they can continue to contract before they fatigue. This is influenced by muscle fiber type, load, and recruitment. So 3 types of muscle fibers are named on the speed of contraction and if they use anaerobic or aerobic pathways. They include slow oxidative fibers, fast oxidative fibers, and fast glycolytic fibers. Most muscles have a mixture of types. There's not one dominant type. Slow oxidative fibers are going to be slow to contract. So what this means is that the myosin is going to move more slowly. Oxidative means that the aerobic respiration will be the primary pathway for ATP synthesis. It will thus have a high myoglobin content, high glycogen stores, it will recruit first, and it will be slow to fatigue. So slow oxidative fibers will be best suited for endurance type activities like running a marathon or, jogging, walking, hiking, swimming, or maintaining posture. And I guess it's a slow walk because as we can see here with fast oxidative fibers, these are going to have fast speeds of contraction, meaning that the myosin is going to move quickly. The primary pathway for ATP synthesis will still be aerobic. There is going to be some anaerobic glycolysis though. There's going to be a lot of myoglobin, intermediate glycogen stores. They're second to recruit, and their rate of fatigue is intermediate or moderately fatigue resistant. And so fast oxidative fibers are going to be used in sprinting and fast walking, not just a stroll, if you would. And then we have fast glycolytic fibers, and the speed of contraction will be very fast. Myosin is moving fast. This is going to be glycolytic, so it's using anaerobic glycolysis, low myoglobin content, high glycogen stores. It's the third recruitment order, and it fatigues very quickly. And so fast glycolytic fibers are best suited for short-term intense or powerful movements like hitting a baseball. And then if you look at the very bottom of the table, you can see that slow oxidative and fast oxidative fibers are going to go from red to pink because they do have blood flow, because they're bringing in, oxygen and lots of glycogen and such to the area. Whereas glycolytic fibers are going to be white or pale in their appearance because they're going to be engaged when blood flow is at its minimum. Remember, the muscles are contracting and it's cutting off blood supply. So there's not as much blood flow there, so they're going to look pale. And you can see that the slow oxidative fibers are the slowest to contract, and they're going to use the most oxygen, and they're going to be useful for those long distance, and postural exercises and maintenance, and they're going to get the most blood flow, so they're the darkest in color. Fast oxidative is going to be used, for quicker movements like sprinting and fast walking and things like that. They're still going to be able to use oxygen though, and so there's going to be blood supply coming in. So they're not as dark as the slow oxidative fibers, but they're going to be like pinker color. And then fast glycolytic, which is like the type 3, is going to be pale in its color because it's going to be lacking that blood flow that is coming in. That's that third recruitment order. So as far as the effects of exercise are concerned, aerobic exercise will lead to increased muscle capillaries, so increased blood flow coming to the muscles, an increase in the number of mitochondria because they're making more ATP, and an increase in myoglobin synthesis. So if you're confused what myoglobin is, I've mentioned it a lot. That's how the muscles will store their glycogen, basically, and also oxygen. So more particularly, the myoglobin is going to be storing that oxygen, because that's going to be akin to hemoglobin in the blood. So aerobic exercise will also result in cardiopulmonary health, greater endurance, strength, and resistance to fatigue. It may even convert fast glycolytic fibers into fast oxidative fibers so that it can use oxygen more efficiently. Resistance exercise is typically anaerobic, and it will result in muscle hypertrophy due to an increase in fiber size, particularly fast glycolytic fibers. And so this one can convert fast oxidative fibers to fast glycolytic fibers. When your muscles increase in their size, you're not actually gaining more muscle cells, but you are increasing the number of proteins in that fiber as well as connective tissue. You're also increasing your mitochondria, myofilaments, and then your glycogen stores. The overload principle is very important when participating in an exercise regime, because you're forcing a muscle to work hard, promoting increased muscle strength and endurance. And the muscles will adapt to the increased demands. Therefore, muscles must be overloaded. You must continue to increase those demands to produce further gains. Paralysis occurs when the nerve supply to a muscle is destroyed and the muscle is no longer stimulated. It then loses its tone and becomes paralyzed. There are two kinds of paralysis. There's flaccid, which is where the muscle becomes soft and flabby, and then the muscle can atrophy, meaning that it wastes away. During development, the muscle tissue is going to develop from embryonic myoblasts that fuse together to form those multinucleated skeletal muscle cells. The growth factor agrin stimulates the clustering of acetylcholine receptors at the neuromuscular junctions. Cardiac and skeletal muscle become amitotic, meaning they no longer undergo mitosis. They can lengthen and thicken, but they're not going to be able to regenerate if they die. Myoblasts, like skeletal muscle cells, will have limited regenerative activity that help to maintain the integrity and health of the muscle fiber. Injured heart muscle is mostly replaced by connective tissue. Now, smooth muscle is different from skeletal and cardiac muscle in that it can regenerate throughout life. It can actually increase in its size. It can hypertrophy by increasing the number of cells during pregnancy, for instance. The uterine walls, which are smooth muscle, will drastically increase. Muscular development will reflect neuromuscular coordination. So development will occur from head to toe and proximal to distal. Peak natural neural control will occur by mid-adolescence. Athletics and training can improve neuromuscular control. In a female, the skeletal muscle mass will make up about 36%. In a male, skeletal muscle mass will make up about 42%. And this is primarily due to higher testosterone levels in the male versus the female. Body strength per unit muscle mass is the same in both sexes. It's just that the males tend to have more of it than females, which gives them that competitive advantage. With age, connective tissue increases and muscle fibers will decrease, and this can lead to what's called sarcopenia, which is the loss of muscle mass. Regular exercise can help to reverse sarcopenia. Arteriosclerosis may block distal arteries, leading to intermittent claudication and severe pain in the leg muscles. And like peripheral arterial disease and stuff like that can affect it. And then here we see our finishing mark. You did it. Thanks for listening.